is Yong Soo. I'm from University of Hawaii, and then I'm at the Department of Human Nutrition and Food and Animal Science. And then it's kind of we had three different departments that joined together. So, and then I'm at the animal science part. So, my presentation is going to be basically related to animal science. Okay, my title is basically production of bioactive myostain propeptide of many animal species in E. coli. So I guess very few of you probably heard about this myostatin. Okay, so let me start with some introduction about this myostatin. Okay, myostatin was first identified in 1997 by Dr. Lee in Johns Hopkins University, and then it is a kind of one member of the so-called TGF superfamily of growth and differentiation proteins. And it's also called, initially it's called GDF8. That's the initial name. And then this is actually the picture from the first publication. What they found out was some genes closely related to one of this family member. So they named it's a GDF8, but they didn't know the function. What's the physiological function of this molecule? So what they did was that they knocked out this gene basically in mice. And then what they found out was this kind of super muscly animal, almost more than 2 times of the muscle mass. So now you can see that the function is basically suppressing muscle growth and differentiation. So they named it as myostatin. Myo means that the flash, and statin is just stopping. So they just indicating that it's suppressing muscle growth and differentiation. So suppressing the muscle mass. And then later, there are many sometimes super muscly animals like that, like in a dog and cattle. Also in humans, we found this types of phenotypic, animal species. And then they found this is caused by the non-functional mutation of myostatin. Okay, so now you know that, right? Myostatin is basically a suppressor of muscle growth. So now we see that I'm in animal science, particularly my area was muscle biology and muscle biology area. So I was interested in how to increase the muscle mass in animal body, because in agriculture, muscle is the meat. So agriculture in animal science, we're handling the production of the meat. So we try to improve the efficiency of animal muscle mass in animals. So I think you can now, anybody here can now think of so if we suppress the myostatin activity in muscles, if there's a chance that we can make them much bigger muscles in livestock animals such as pig or poultry. So that's the kind of strategy. So let's think about the strategy. What kind of strategy available? I think one is that the genetic approach. I think this is one of the popular. We can utilizing different types of genetic approach, such as transgenesis and siRNA technology and transgene transfection. All this kind of utilizing can utilize this strategy to suppress myostatin activity and then increase the muscle mass. And then actually people did. And then this is the wild type animal, and then this is the dominant negative activin receptor 2B. Activin receptor 2B is the myostatin receptor for its signaling. So they make it they suppress this receptor. So myostatin cannot bind to its receptor. So myostatin activity is basically suppressed. So when they create this transgenic animal, as you can see, there's an increase of muscle mass, right? This is like a follistatin overexpressing transgenic mice. So follistatin is a molecule which binds to myostatin and suppresses activity. So when they produce this transgenic animal expressing abundant amount of follistatin, you can see there's a huge increase of muscle mass, right? But for me, this is in terms of the study, this is a lot of interesting studies you can design here. But for me, I'm in livestock. I'm in animal science. So, anybody know that any country ever approved the use of transgenic animal for human consumption of the meat from these transgenic animals? No country ever approved that. So for me, it's a little bit of a dilemma, right? So because I'm really interested in making muscle mass bigger using suppressing this myostatin. So I think basically alternative strategies is protein administration. You can produce antibody against myostatin, or you can find out the myostatin suppressor, such as propeptide, activin receptor 2B, and follistatin, some other molecules. We can just produce this one and then maybe try inject or administer these proteins to the animals and then suppress the myostatin activity, and then hopefully resulting in a muscle mass increase in these animals. So I tried antibodies. I tried antibody against myostatin, and then I used in my study I used injected to the fertilized chicken embryo, and then turned out increasing muscle mass. And then I kind of switched my project to producing these types of proteins. So basically, I'm going to talk about now the production of this protein in E. coli. So the two proteins here, myostatin propeptide and follistatin. I think if I have enough time, I'll probably talk about a little bit about the follistatin, but the main focus is going to be this myostatin propeptide. So the question is that there's nothing new here, right? Everybody producing recombinant technology to produce proteins. There's nothing new, right? And so this protein is also commercially available, and then some labs they produce this in eukaryotic system. As you know, eukaryotic system is costly. It's very expensive. For me, I need a large amount of proteins to actually do an animal study, like injecting the pigs or even cattle or poultry. So quick look at the price of this. This is a commercially available molecule, propeptide and follistatin. So 25 micrograms around $329. Maybe if you bulk order, you can make some deals to make cost may go down a little bit, but not much, I guess. So think about it. So just a simple mouse study, you're going to inject. So this is the assumption. 6 animals for injection, and then using 100 milligrams per kilograms. But this is a one experiment, okay? Then I need about 2,400 micrograms, 2.4 milligrams of protein for one simple mouse study, right? It's going to cost about $31,000. Not much, actually. We can afford to this much of money to buy these proteins. How about the pig study? A pig study, 6 animals, 2 injections, same dose. The calculation comes out more than 200 milligrams. So if you buy this from the commercially, it's going to be $2.68 million. So anybody, if I propose this study, anybody can support this type of study? Obviously not, right? USDA or NSF or any private funder, they're not going to just support this much to simply buy these proteins, right? So I need to make this more cost-effective system. So I try to use an E. coli system. But there are some previous attempts to produce a propeptide in like E. coli. What happened was that it's all produced as a inclusion body, not soluble forms. Okay, it's an inclusion body, so you have to collect the proteins, and then later you have to solubilize and then folding, right? There's a known like a protocol for perfect folding of the proteins. A lot of times these are just trial and error. So I try to avoid this folding process because in a long time ago I tried to produce a myostatin molecule itself in E. coli. Then I just 3 years, 4 years, I couldn't make any single publication because we tried all different kinds of folding process, never folded. So I try to avoid that. So also follistatin, follistatin, some group tried, but they failed. Follistatin is very, very complex molecule, so it's not easy to produce in E. coli. Okay, so what before talking about the system, I'm going to just talk about a little bit about myostatin propeptide. By the way, myostatin is like translated into into a pre-propeptide. Okay? So they have a signal sequence there, and then when going to the endoplasmic ER, then it's removed, and the inside the ER and the Golgis, they're going to be processed. So it's going to be cut here at this site, and then they separate this is active myostatin, and then this is called propeptide or prodomain, right? The interesting thing is that after, by the way, this myostatin forms a dimer like that, forms a dimer. After cleavage of this propeptide, they're making a complex with these active forms of myostatin, and then they suppress the bioactivity. Okay, that's one of the chemical characteristics of this prodomain. So the thing is that for this myostatin being activated, binding to its receptor, it should be like a processed. And then the current understanding is that there are proteins, metalloproteins called BMP1 toluene metalloprotein. Then you cut like a 99 position of aspartic acid, and then you cleave, and then it's going to be separated, and then myostatin becomes active. So for this prodomain production, maybe making a mutation in this site is going to be much more effective, because it's not going to, when you're injecting to the animal, it's not going to be cleaved, right? So we just made a wild type and also the mutant forms of this propeptide. And then this is the system we use, and then we tried a different kind of a fusion partner. We failed. Then we found out this maltose binding protein being used as a fusion partner in making the propeptide soluble in E. coli. So we used this system. It's called PMA C5. Nothing special here. You just have an MVP as a fusion partner. When you're expressing it, the MVP going to be attached to the N-terminal side of your protein. So we use this one. The other thing is that this MVP can be removed later. So there is like a cleavage site between the MVP and your protein can be removed by the factor XA protease. So you can remove it after production. You can remove it later. So just a simple this cloning process. Then produce. So if you're looking at it, this is the wild type mutant, basically the same, and then we induced, and then we after cell lysis, look at the solubility of this protein, and then this is a supernatant. So you centrifuge and then cell lysate, and then this is the total. So almost more than like 90% is all soluble. So at least we successful in making this soluble. Okay, the next one, and then we do simple affinity chromatography. Maltose binding can be used affinity tag using the amylose. Oh, sorry, the typo here. Using the amylose affinity chromatography. So we do some purification, but there are still a lot of other proteins. So next one is that basically we did ion exchange here. So you can see that a little bit, increase the purity, but at least these smaller ones just removed, but still the bigger ones in there. So we did two purification process. And then the other one is that basically, I told that it can be cleaved by the factor XA to separate the MVP and the protein. And we tested this, so factor XA presence, we can see this one becomes the two major band here, so indicating it's being separated. You can cleave it, and then so this is the our protein, and this is our MVP basically. So we did Western blot. We confirmed that actually this we can separate this from the MVP. Okay, next is just a simple characterization, biochemical characterization of the binding, and then also the other one is that we needed to confirm actually this mutant is really mutant, so it's resisting this protease. So do the BMP1 digestion like this. So here, this is in the presence of BMP1, and then without BMP1 on this lane here. So you can see that this is a wild type is cut two band here, but mutant is not cut. So it means that the mutant was real mutant, just resisting the BMP1. Okay, and then we did also like a pull down assay to see whether it is really binding to myostatin, and then this is the pull down incubating the myostatin. Here myostatin band is about 12.5 kilodalton, and then this is our proteins, and then this is the MVP. So when you mix with the MVP, myostatin is not binding. So this is a negative control. And then we found that it's coming together, indicating that myostatin really binding to our recombinant proteins. Okay. And then we did in vitro bioactivity using the this luciferase in the system. The system is basically supposed to, when you add a myostatin, it's supposed to produce a luciferase. So when you mixing the myostatin, this protein together, it's going to suppress. If it's really, really active, it suppresses the myostatin binding to its receptor. So the luciferase activity should be suppressed. So you can see that here. So we, with the increase of concentration, the activities inhibition is 100%, and then with the decrease of this ligand of our recombinant proteins, is, inhibition is decreasing like that. So we used a commercial, the eukaryotic produced myostatin prodomain as a control, and then we compare our recombinant with this one, and then basically the pattern is pretty similar. If you calculate the IC50, IC50 is just no statistical difference. So indicating that activity is almost same as the eukaryotic produced myostatin prodomain. Okay. So and this is the yield. So basically, from the 1 liter culture, we can produce like a 38.7 after affinity chromatography, ion exchange a little bit reduced after removing this MVP. It's about 4.2. So at least we can produce around 4 milligrams per liter. Okay, that's the first result. And then let me see. The other one is that then after this pig, we also produced different species of prodomain like pig and chicken. We even tried in fish, and we found that their potency is quite different. You can see that. This is a fish here, and then this is the ligand concentration. So this is getting higher, and then this percent inhibition. So if the ligand is higher, it's going to be suppressing myostatin activity, right? So if the graph goes to the left side, it means that it's more potent. So this fish is here, and then chicken is here, and then these two are the mouse and then pig myostatin, and then basically the same. So we found out different species, this potency is a little different. If you're looking at it, the fish is almost tenfold higher than either mouse or pig prodomain. So we found that just differences in different. Okay, after that, we kind of try to truncate. So try to find out what's the minimum size of the prodomain. So we're making this different 42 to 18, 42, 175, 42, 115, 42, 98. And then the other also goal was that put the like FC fusion proteins, because it's going to increase the potent half-life, right? So we try to add that. So we did the same thing, and then I'm going to show that. So we produced it, and then these are the molecules, and then these are the yield of this different truncated forms of prodomain. So if you're looking at it, smaller size, we had a much higher production than the bigger size. So this is a good news. So if the activity is same, if you're making like a small size, it's going to have a more yield. And then we'll measure the bioactivity, and this is the basically bioactivity. So up to 175, the activity didn't change much. But after that, 40 to 115, the potencies are decreasing, 40 to 98 even further decreasing. So we found that this is the kind of a minimum size to maintaining their potency. Okay, now this is kind of interesting. Now I said that this one for activation of this prodomain, this cut here, just 99. So this is actually here. So this is supposed to be this side. It should not bind myostatin, so it should have no activity. So the reason we did this was that this one's supposed to have no bioactivity, but what we found out was, it has a bioactivity actually. It's a lot of bioactivity. So we still don't know, what's going on, but looks like MVP may have some play. Even MVP binding may be stabilizing binding to the myostatin. So that's just our guess. Okay. The reason I put this was that there are some, actually 2004, they did some, Xiang's group did some study, and then they also created 40 to 98. This is mouse, not pig. And then they didn't find any activity at all. But for our case, we found activity. And then the only difference is that the fusion partner. So it looks like the fusion partner was something about this. And then the next one is that just F fusion. Okay, so we did this, and then same thing, we produced amylose affinity purification, and then we can for the purify using protein A, so we just purified for the purification, and then here's the three proteins. And then measure the activity, kind of interesting. If you're looking at it, actually FCR fusion improve the potency of these proteins. Commercial is even higher than the commercial protein. Another interesting thing was that this 40 to 175 is without the FC, it's almost same as this non-truncated form of propeptide. But now it goes potency decreasing, and then the potency is kind of lower than this 40 to 115. So when you attach the FCR, the C-terminal side, actually the activity stays the same. So that's kind of a little interesting. So we kind of now focusing production of this one too for animal study. So this is the yield. So if you're looking at it, the yield is this is much higher than like the other two the sequence. So that's what we found. So we're focusing production of this one right now. I think I'm going to use 1 more minute and then just finish up. Okay, this is a follistatin. Follistatin is a very, very complex molecule. It has disulfide bond. There are 5 different domains, and then there are more than like 10, 15 disulfide bond information. And then this is a structure of follistatin, different domains. And then what we tried was that because of a lot of disulfide bond, we initially tried this periplasmic expression, and then it failed. When it did that, it's all like this not soluble form. So what we did was that there was something called shuffle strain. What we found was when we used the shuffle strain, actually it expressed as a soluble forms, and then we could purify using the amyloid and heparin affinity purification. So up to this much of purity. And then this is the activity. And then the things I like to point out is that basically the activity is kind of compared to the wild type is much lower. The potency is much lower. But also for the activin, it's also potency is much lower. That's what we found. But at least the good thing is that we first time we were able to produce the follistatin in E. coli actually. So okay, so concluding my presentation, basically we were able to produce the propeptide and then also follistatin in E. coli system. So having this now allows us to do actually animal injection study. So we are in the process of doing animal, particularly in the pigs. So we're injecting this newborn pigs and then see whether they're affecting this growth of these pigs. That's what we're doing right now. Okay, so there are some acknowledgement. There's my collaborator for this project, Dr. Bang, and then Frankenstein, and James, and Cho. There's some of my students involved in this project. Okay, thank you. At least how much time for a question, I guess. Maybe just one question, I guess. Okay, great. So let's move to the